Hello, thank you all for joining us at the MID 2023 Clinical Track Program. My name is Tom Sun, VP of Strategic Consulting Services at Cognigen Simulation Plus. It is my pleasure to introduce our speaker, Professor Manuel Ibarra, who is a professor at the University of Public Uruguay as our speaker. And his topic for this talk is towards a flexible model-informed precision dosing software solution using Monolix suite. And before the presentation, I would like to share a couple of housekeeping remarks. First of all, we treat your privacy rights very seriously by participating in this conference or provide Q&As. You agree for us to contact you in the future for follow-up. Secondly is that please use the Q&A tab on your dashboard for any question at any time. If you have any technical issues, please use the chat to type your question. Our staff will try to help you. Without further ado, Manuel, here's for your presentation. Thank you.
Thank you very much, Tom. Hello to everyone. It's very, very nice to be here. I want to start thanking the organizing committee for inviting us and having the opportunity to be here with you all, sharing a bit of how has been our experience in bringing pharmacometrics into the clinical setting here in Uruguay, where of course I will give you a bit of context, but as you might know, pharmacometrics is very, very incipient. I will be talking on behalf of my colleague Laura Gonzalez, who could not be here today, but we are both part of an interdisciplinary group, which I will comment further on. So to give you a bit of background, we are here in Uruguay, in South America, in the city of Montevideo. I work at the public university, which is the only university of the country. I don't know if you know, but Uruguay is a small country with 3.5 million people, population. And we work right here at the university hospital, which is right beside the Centenarium Stadium, which is very popular for being the first place where a football World Cup took place in 1930. So right here in the University Hospital, we work together within a therapeutic drug monitoring unit and giving other service for such as biopharmaceutics and so on. But today I will focus on what we've been doing in model-informed precision dosing. So to bring us all on board, just a quick some words about precision dosing. What we are talking here is tailoring treatments to patients' needs. So precision dosing involves individualized drug treatments based on patient characteristics, which are known to alter drug absorption, disposition, and response, with the aim of maximizing the probability of attaining both goals, efficacy and safety. And this is opposed to the flat dose or one-size-fits-all approach, which could be considered as a traditional approach. And if we know, because of our clinical experience, that a few drugs works well in all patients under the same dose regime. So there's a really few works that you apply the same dose regime and you can attain both goals, efficacy and safety. So this is about delivering the right dose, the right drug in the right dose to the right patient at the right time, and it should be placed or presented as a cornerstone of precision medicine, which is the optimization of disease prevention, diagnostic, and treatment strategies by targeting the needs of individual patients on the basis of genetic, biomarkers, phenotypic, physiological characteristics. And that is why we call it precision dosing, because we want to be partners in this precision medicine. Although, well, we are talking about dosage regime, it could be maybe more suitable to talk about accuracy. So when we combine precision dosing and quantitative pharmacology or pharmacometrics, we get model-informed precision dosing, which is use model and simulation approaches in combination with individual measured patient and disease characteristics to inform clinical decisions within the context of precision dosing, or could be precision medicine. So we basically use pharmacometric models that we can develop on site, or we can use published available pharmacometric models, which can be populational PK/PD models, or it can also be PK models, for instance. And we use those models to give a priori predictions, and also to get individual estimates, as we will see in some slides. These models should be validated, and there's a lot of background behind the implementation of pharmacometrics in this context, which I will briefly cover. But basically, that's the idea. So model-informed precision dosing is really key to bridge knowledge. As you know, for the post-marketing stage, the dosage recommendations are based on the clinical studies involving relatively homogeneous population, because of the clinical trials involves several inclusion-exclusion criteria. So then we need tools to mechanistically bridge that knowledge into the real world, real patients with a lot of the variability is much greater, and we have different special populations that were not studied before. So quantitative pharmacology can put that in place and give us the tools we need to achieve earlier to more safe to efficacy and safe dosage regimens. So the most common method that we use in model-informed precision dosing is the use of nonlinear mixed effect models, which involves a set of parameters, structural parameters, defining a PK model, defining the longitudinal variability. So this is concentrations versus times, or effect versus time. And then we have random effects, defined distributions for the inter-individual, inter-location, and residual unexplained variability. So a nonlinear mixed effect model is really a joint distribution that provides an a priori prediction based on typical distributions, and can be combined with patients' data to obtain an a posteriori distributions and predictions for a given subject. So we can use an LME model combined with covariate data for a patient to give a first a priori prediction for the drug exposure relationship. And then we can use this these models to combine with observations that we can obtain on a given patient to estimate individual parameters, and then provide a sort of individualized prediction for a given patient, based not only on their covariates, but also on what we are observing on the drug exposure and effect data. So how we do that? How this is done? We use this mostly the most common method is using maximum a posteriori estimation, which balances prior knowledge, which is the model, the joint distribution that is included in a population PK or population PK/PD model for a given population. And this is balanced with individual data to get an estimate of an individual parameter. So as you can see here in this example, so we have a model that gives a prediction, an a priori prediction, which is here shown in blue, for a patient based on covariates such as body weight, sex, or for instance, creatinine clearance. And then we have the data here, it's a simulation, the data collected in a patient. Okay, so the a priori prediction will be this one, the blue one. And then we made observations in the patient, which are shown here as red dots. And the a posteriori prediction, so the result of getting individual estimates using maximum a posteriori estimation is shown here in red. So as you see, if we only consider individual data, we will get the green curve which fits very, very nicely the individual observations. However, we will be ignoring what we know about the population for which we assume that the patient belongs to. So it's really important then to have a way to integrate this information, because it's knowledge that we have and that we should use. And for instance, if you take a look at the absorption phase, there's only 1 or 2 observations that are telling us that this patient has a much slower absorption compared to the typical prediction. And this absorption is only based on 2 observations, which could have experimental error. So the a posteriori prediction balances this 2 sources of information to get us this a posteriori curve, which, as you can see, is near to the typical prediction for the absorption phase, but then is near to the individual prediction, into the individual fit in the disposition phase, where we have more data for this patient. Of course, if we gather more data and we can experimentally prove that this patient has a really slow absorption, then the a posteriori prediction starts to tend to the individual fit. So this is how we balance the data and how we use the models in the clinical setting. We could call a Bayesian updating and Bayesian forecasting. So if we have for a new patient, we have no observations. We only have data of covariate characteristics, such as here is reflected with body weight. So for instance, we have a patient of 60 kilograms, then we will make up our predictions based on this distribution for the clearance. And as we gather more observations in this patient, we can refine our individual distribution for this given patient, and this should give a more precise estimations and more precise predictions and more accurate predictions for the pharmacokinetics of a given patient. So what we are doing here is individualizing a POP-PK or POP-PK-PD model. And so this is how it is done. We have to perform a Bayesian update, and then we have to make a prediction based on that new individual model. So this is kind of a complicated and time-consuming task, relatively, probably not for an experienced pharmacometrician, but it is for pharmacists or which are made, or clinicians which are not involved with pharmacometrics and want to implement this in the clinical setting. We have, as soon as we have individual observations, we have to create a data set, estimate individual parameters, perform simulations, and make a report. And this is a cycle that it's renewed every time we have more observations. Very important. MIPD works, or at least the studies, there are different reports studying how this performs opposed to a traditional approach. And this is a very nice work by the group of Mike Neely, in which they implement MIPD program, and they compare. So during the first year for vancomycin, they did not follow, so they use only recommendations based on the physician criteria. On the second year, they used MIPD, so they used model to inform the dose decision. And at the third year, they did the same, but they perform an optimal design to improve the experimental data. And as you can see here, so vancomycin is the target is an AUC over the MIC, over the MIC. And as you can see, for year 2 and year 3, where models are used, the AUC are stretched or they are more close to the target AUC, in contrast with the first year. So, and to put this on words, so the Bayesian AUC guide dose in years 2 and 3 was associated with fewer additional blood samples, shorter therapy durations, reduced nephrotoxicity. Although the efficacy was similar, this is a really an important advantage for patients and for the healthcare provider. And there are other reports, such as this as Roggeveen, where the same observations were made. So there are clinical benefits, improving patient outcome, and there's also a financial impact that we should promote. We should discuss this with healthcare authorities, as we know that the implementation of MIPD can reduce the incidence of preventable adverse drug reactions, improve adherence, reduce hospitalization days. Of course, there is a lot of challenges here. This is a very nice review by Chloe and co-workers. There are a lot of the underlying challenges which involve practical aspects, scientific aspects, understanding the value of MIPD in research setting and routine clinical care, a common definition which is very challenging as we get on board of interdisciplinary field of application. And then we have the technical. Our goal is to implement a model which can be connected, can integrate data to inform individual dose regime. There are great software available, and this is also a very recommendable review, which provides a lot of solutions. In this case, I will talk a little about our experience. So here in Uruguay, we have some difficulties to implement and to use these models, and also we have other kind of needs or perspectives. Although I have to underline that the groups behind or developing the softwares I showed are really pushing forward and leading the advance of the field. And they're doing this is very, very nice work, really advancing the field. So here in Uruguay, we have issues with software accessibility in terms of costs. We need to engage medical staff and hospital authorities in a context where the use of modeling and simulation is not popular, or they simply have no idea what this is. We need the ability to apply the models that best fit our needs and perspectives. So the models that best fit our population, and many times these are special populations, and we have the need to start off on the right foot in terms of model performance, because as this is new, it's important to convince and to show that this can really fulfill the objectives that we promise in the first place. So those are some needs we have, and there are things that any pharmacometrician applying MIPD should be aware. And one key aspect is intermodal variability. So as I mentioned before, you can develop your own model with your own population, and that would be great, but that is very time consuming, resource consuming, and it's very likely that you don't have the time to develop a POP-PK model for all the drugs that maybe you can start performing MIPD with. So this is so a lot of times we could take a model which is published in the literature and start using that model to recommend dose regression in our population. But we have to be aware that there's a lot of variability in the predictions of the different models. This paper shows that by Broker and co-workers for vancomycin. This is 1 and 2 compartment models that are reported, and as you can see, the variability is very large. We have studied the same with tacrolimus. We evaluated nearly 30 models, different models, and we have seen a lot of variability there. This phenomenon makes it makes that you have to validate the model. So you have to qualify the model with your own population before starting to use that model in an MIPD contest. So we mainly evaluate accuracy and precision. And here you can see in the same review for different models of vancomycin that this accuracy in prediction is evaluated for the a priori prediction and for a posteriori prediction at different occasions in which more data is being gathered for each patient. So as you can see, that the prediction and the precision and accuracy both are improving with more data for each patient, but there's a lot of variability for different models. We did the same for tacrolimus in our population, and we see that these are prediction errors summarized here as box plot. And as you can see, there in our population of 40 renal transplant patients, there are very few that have good results, and the variability is definitely very large. We did the same with anti-tuberculosis drugs such as isoniazid and rifampicin, and same results. And with vancomycin in critically ill patients, we here have better results, but the message here is that you need to do this because intermodal variability is definitely something that is there. So as model-informed precision dosing is advancing to the clinical setting, there's a need for flexible software solutions that support its application by pharmacometric groups, because pharmacometric groups on site can do a lot to push forward the field and the application. But they need to have enough flexibility to implement their own models or any model they might want to use to contribute to demonstrate the advantage of the approach in the clinical practice and clinical research. In our experience, we have gathered with other disciplines, pharmacists, physicians, veterinary doctors, statisticians, and computer engineers. We, from here at the university, this is an interdisciplinary model-informed precision dosing group, which has been working for more than a year, and we have had great experiences working with these groups. And I will within this group, and I will show you one application we developed to fulfill some of the needs that we have. So the software solution we developed. I will show a prototype, and there was some needs that we wanted this tool to have. So we want to be accessible, to be cloud-based, having a server and client architecture. We wanted to be user-friendly with 3 modules, one for physicians, which could be able to simulate from the population parameters. So a physician should be able to perform the a priori prediction when they face to a new patient. And of course, a physician will not be involved with the Bayesian updating and Bayesian forecasting, but they should be able to simulate from the population parameters. Then another model for pharmacists at the TDM unit, able to perform the Bayesian updating and forecasting and perform the recommendation to the physician, and an administrator which is able to change the models and platform structure. This other characteristics store individual data, able to integrate new NLME models. So any model we already have developed, we want it to be automatically and quickly integrated into this tool to be at the next day implemented in model-informed precision dosing. So provide PK metrics of interest and generate reports. So we work with a group of computer engineering, and we came out with a prototype that it looked very nice, and I will show you in a bit. So this is the architecture. It has a front end with an administration panel and application, and at the back end, this works with Monolix and Simulix. So any model at the moment written in Simulix, in Monolix, can be integrated and started to use Monolix to estimate and Simulix to perform the simulation. So the tool allows to create a data set, estimate individual parameters, and perform simulation and perform a report. And I will show you a demo. Let me just go here. So, of the tool, that's the the main page. The tool asks you to register. This is for admin for different premises to access the different models. So the first thing we have to need is to register a new patient. That's a popular football player, Uruguayan, Diego Forlan. This right now the tool will perform a priori prediction. We can select any model that is integrated into the tool and include the covariates for the patient. So covariate data that of course depends on how the model is built. So different models will ask you here in this place different covariates. So after that, we create different treatments, and we can create any treatment we want to perform the typical simulations, as you can see there. Then it gives you a brief. Then it uses Simulix to perform the simulation based on the POP-PK model that is integrated in the tool. It provides very nice graphics that you can. This is built on React, so you can play around with this graph, and you can zoom in or have a sense of how the system will evolve under the different dosage regimes, and also gives you important PK metrics that are important to assess to assess in the right way to the physician, such that the time to reach the steady state, the AUC, the maximum concentration, minimum concentration. Then you can generate a report, which is very basic at the moment, but it generates a report in PDF, and you can share that with a physician. Then you can input data for the patient. So if you already have a patient registered, so there's a data set for the different patients here hosted in the server. You can load different here. You load more characteristics of the patient, and then you load the observations and the treatments that the patient was following. That's what it's doing. You can upload as many observations as you collected. And this is to perform the Bayesian updating process. So as soon as you add different observations and you save this, so the tool will be communicating with Monolix to perform the estimation, the maximum a posteriori estimation of individual parameters. And then you go back, you choose the patient, and you can now simulate different treatments, but using the individual parameters. So basically, what any model, any model, any tool, any MIPD tool could do is not that complex. I think that the most valuable thing of this tool comes here. It has an administration panel that allows us to integrate any model we have developed. It can be a PBPK model built right now in Monolix. So you integrate and you integrate the MLX code here, and you integrate the values of the different parameters, typical parameters, and then you're ready to use that model to perform MIPD. And there's a data set. So for sake of time, I will jump to I have two more slides. So we have next steps, because this is a prototype and we want to keep on improving this. There's a lot of things that we can still improve, and we are working on this. And to close, some takeaways that we have experienced. Interdisciplinarity is essential for MIPD success. We have had a great experience working with other disciplines. There are several challenges to be solved with sites, so it's important for pharmacometric groups working at the hospitals to be working on this and promoting this together with other disciplines. There's a need to prove the medical staff and healthcare center authorities the benefit of MIPD, and there's a great software available And also pharmacometricians might want to use their own validated models, and that's where our prototype could be useful for other groups. So with that, thank you for your attention. I know we are very close to the end. Tom, thank you, and I will be happy to take questions at the Q&A session later.
Great. Thank you, Manuel. And just a reminder for our audience, we have now the section 10 after 2 o'clock Eastern Time, US, for the Q&A. And so please come and interact with the Manuel at the time. But I want to thank all of you to participate, and thank you for speaker for your insightful presentation. So we're going to end this session now. Thank you.
Bye-bye.
Bye.